Hello and welcome to the second and final day of our National Alzheimer's Coordinating Center 2021 Spring ADRC meeting. Today's research education component session will be from 10 to 11 a.m. Eastern, but without further ado, I'd love to pass it off to Elizabeth Head from the University of California, Irvine. She's one of our REC steering committee members. Take it away, Liz.
Thanks, Anna. Good morning, everybody. I know it's early for us West Coasters, so I'll just give you some brief updates on what's going to happen on the next session. Next slide, please, Hannah. So our session today is going to include the second set of presentations, nominations from each ADRC for their REC trainee. So yesterday's session was absolutely fabulous, so I expect no less than that today. If you're interested, all the submissions are available on a box, which we'll put the link in the REC channel as well as on chat if you would like to go and see those. And in the end, we will house all of these presentations on the REC website. We're going to ask our presenters to provide a photograph for the website as well. Not everybody recorded Zooms with their pictures in there, so that when we see each other in real life, which hopefully will be soon, you'll recognize each other in person. There'll be a link we'll put into the chat so that you can vote for your top 2 presentations today. And we've tried to balance for East Coast, West Coast. And we anticipate closing our voting at 12 p.m. And the excitement of the day will be around 1:45 Eastern time at the directors meeting where we're going to announce the winners. So we're pretty excited about this. Next slide, Hannah. So here's a list of our day 2 presenters. We have representatives from Emory, University of Kentucky, MGH, Indiana, Knight, the University of Pittsburgh, Rush, Columbia, Yale, and Michigan. So some pretty outstanding speakers and panel that we anticipate today. So with no further ado, let's start our wonderful presentations.
Hi, my name is Lena Higginbotham. I'm an assistant professor at Emory University. I'm part of a multidisciplinary proteomics laboratory, and today I'm going to talk to you about how we identified network-based CSF biomarker panels in Alzheimer's disease. So a little bit of background, increasing evidence indicates that AD pathophysiology, of course, extends well beyond hallmark amyloid and tau accumulation. In particular, there appears to be a complex decades-long cellular phase of AD involving a dysfunction or variety of cell types that contributes to irreversible damage. And core AD biomarkers, was primarily focused on A-beta and tau recognition, currently poorly reflect this diverse pathophysiology. So since we are a lab that really specializes in unbiased network-based proteomic analysis, our goal has been to develop these protein biomarker assays that fully reflect the diverse disease pathophysiology of AD. Over the last several years, we have been working diligently to define the AD brain network proteome. And today I'm going to tell you about how for the first time we integrated this AD brain network proteome with a differentially expressed CSF proteome to identify fluid-based biomarkers. For those of you who are not familiar with network proteomic analysis, it's really a cutting-edge way of organizing complex proteomic data, such as what you find in the human brain, into groups or modules of proteins with similar expression patterns across cases. And oftentimes these modules reflect particular cell types or functions or organelles, and therefore it can give you a really global picture of disease physiology. And again, this project, for the first time, we integrated it with the CSF. So we used 2 different proteomic data sets here. We used a deep CSF proteomic data set involving 3,000 proteins, over 500 of which were significantly altered in disease. And we used a really deep brain proteome, over 8,000 proteins, 7,000 of which segregated themselves into 44 modules. We then used a Fisher exact test to identify the key or significant points of overlap between these 44 modules and the CSF proteome, including the entire CSF proteome, and then just differentially expressed CSF proteins or potential biomarkers. And we found that there were really 15 modules of interest that really overlap significantly with the CSF proteome. And once we actually collapsed these modules by various features, we found that they represented a vast array of disease pathophysiology. So we had a synaptic panel, vascular panel, myelin, glial immunity panel, and metabolic panel. You can see that they reflected a lot of different cell types and various gene ontology terms. And each of these panels was associated with a fair number of differentially expressed CSF proteins. Once we replicated these panels across various cases, we found that there were 60 highly reproducible, somewhat AD-specific CSF biomarkers across these 5 panels that we really are going to take into the targeted validation stage. And you can see once we whittled down these proteins, it was nice that these panels had a clear directionality in disease. Several of them, 4 out of the 5, were actually going up in disease, as you can see here, while the vascular panel was going down in disease. And we had, we replicated this over 500 plus CSF cases in several different cohorts, and we found a lot of reproducibility here and a little bit of disease specificity, as you can see here with certain panels. And then finally, another key finding was that when we looked at our panels in asymptomatic AD CSF, we found that there were changes already in early disease. And once we took some of these proteins, the top changing proteins, we actually found that these proteins could segregate ASYM-AD into 2 separate groups. One group that looked more control-like, one that looked more AD-like. So this indicates that potentially our panels could risk stratify people in early disease. So this is exciting. So like I said, we are taking this to the next step, currently using targeted MS to validate these various CSF panels. And we're moving, what we hope to is move toward multiplex proteomic assay development. I'd like to thank our entire lab, including Ling Yanping and Eric Dammer, who did a lot of work on this with me. Thank you.
Hello, and thank you to the organizers of the meeting today. My name is Elizabeth Rodas, and I'm a postdoctoral fellow at the University of Kentucky Alzheimer's Disease Research Center, as well as an occupational therapist. My training and clinical experiences have provided unique insights related to functional abilities for people living with cognitive impairment. Behavioral and psychiatric symptoms of dementia create challenges for care. Many BPSD share similarities with pediatric autism spectrum disorder, such as anxiety, depression, repetitive movements, and difficulty with social interactions. Autism has been shown to have similarities to frontotemporal lobe dementia, but comparison to other dementias has yet to be explored. To address this gap, we surveyed informants of individuals with cognitive impairment from the UK ADRC using the Gilliam Autism Rating Scale, or GARS. We followed this with a secondary analysis comparing GARS results with the neuropsychiatric inventory and the CDR sum of boxes. The unique opportunities of the longitudinal and translational research offered at the UK ADRC allowed us to assess post-mortem neuropathologic and neuroanatomic involvement of participants who presented with antemortem autism behaviors. We found that a substantial number of participants did present with autism-like behaviors, and that participants with high autism scores were significantly younger at age of onset by 5 years. They had advanced cognitive impairment compared to those with lower autism scores, and the behaviors were not limited to those with frontotemporal lobe dementia, but were present in a variety of dementia types. Behaviors measured on the GARS and the MPI were correlated, and the GARS added 6% variance to cognitive impairment when controlling for variables such as sex, age, and education. The individuals with autism behaviors scored greater than 1 standard deviation higher on the GARS compared to the individuals with high BPSD but without autism-like behaviors. Neuropathologic analysis found an increase in tau pathology clustered within the frontal and temporal regions of people who presented with autism-like behaviors prior to death. In this image, we can see the individual with autism behaviors in panel A had substantially more tau tangle pathology in the frontal region compared to the parietal region in panel B and the match control in panel C and D. Over the last 5 years, we've used clinical observations of behaviors presented across the lifespan and what were thought to be unrelated neurological conditions. And we've identified the presence of autism behaviors in those with dementia, as well as a relationship to BPSD, but also a different trajectory, including earlier age of onset and advanced cognitive impairment. and a different pathologic substrate than those without autism behaviors. We're now taking this research one step further to develop non-pharmacological interventions to improve treatment options for BPSD. Sensory processing abnormalities are prominent in children with autism and contribute to disruptive behaviors. In a small sample, we identified older adults with cognitive impairment also exhibit decreased sensory processing abilities. We're currently completing a proof of concept clinical trial which provides occupational therapy over telehealth and sends sensory stimulation kits directly to participants' homes to help mitigate BPSD. This is a similar intervention used in pediatric therapy settings for autism. Sensory experiences are based on one's environment. So we're borrowing Kurt Lewin's equation from 1936, which indicates that behavior is the direct function of the person and their environment. This fall, we're partnering with the Emory Roybal Center to embed a sensory-based intervention in the home environment by training caregivers to self-initiate sensory techniques for improved behavior. Finally, I would like to thank the participants and care partners of the UK ADRC, which allowed for this research, as well as all faculty and staff for supporting and training young investigators like myself. I would also like to thank the Emory Roybal Center for the support of the Harmony at Home intervention trial this fall. I would also like to thank the NIA for supporting the UK ADRC and the T32 program, which have been vital for my education and research career.
Hi, my name is Alberto Serrano Poto, and I'm an instructor in neurology at the Massachusetts ADRC and Mass General Brigham. Astrocytes and microglia are known to react to amyloid plaques in the Alzheimer's disease brain, and traditionally this astrocyte and microglia reaction has been depicted with single marker immunohistochemistry, for example with GFAP for reactive astrocytes and with IBA1 or CD68 for activated microglia. However, glial responses are complex, and we hypothesized that a cyclic multiplex fluorescent immunohistochemistry protocol will help us understand this complexity. We have developed such a protocol with up to 8 cycles of immunohistochemistry on the same formalin fixed paraffin embedded section and up to 16 markers or antibodies on the same sections. These include 7 markers of astrocytes, 6 markers of microglia, 1 of neurons, 1 of amyloid plaques, and 1 for tangles. We cropped out more than 5,000 ALDH1L1 astrocytes from 7 controls and AD brains, specifically from the temporal cortex, and analyzed the signal intensity for all the other astrocyte markers. And we found expectedly an increase in the intensity of reactive markers, GFAP, YKL40, and TSPO, to a lesser extent also Vimentin, but no change of the homeostatic markers, glutamine transporters 1 and 2, and glutamine synthetase. But because we had a multidimensional data of signal intensity on thousands of astrocytes, we decided to use spectral clustering to try to discern between astrocytes phenotypes or states. And we actually found 3 broad phenotypes with this method, which we call homeostatic, intermediate, and reactive. The homeostatic astrocytes were majority among the control astrocytes, whereas the reactive astrocytes were majority among the AD astrocytes. By contrast, the intermediate astrocytes were represented by both control and AD astrocytes, and they were characterized by the highest levels of glutamine transporter 2 and glutamine synthetase. Similarly, we annotated and cropped out more than 6,000 IBA1 positive microglial profiles from the same sections and analyzed the signal intensity for all the other microglial markers. And we found that all of them had increased signal intensity in AD versus control microglia. When we applied spectral clustering, we also observed 3 different phenotypes. Again, we termed them homeostatic, intermediate, and reactive. And again, the homeostatic microglia were overrepresented in the control brains, whereas the reactive microglia were majority among the AD microglia, and the intermediate microglia was equally distributed across control and AD brains. Interestingly, this intermediate microglia had the highest levels of TMEM1 and 19 and ferritin. we went ahead and tried to classify AD versus control on the basis of associated microglia image features. To this end, we built convolutional neural networks with these architectures that you can see in the left, specifically 4 convolutional layers for feature extraction and 3 densely connected layers for classification of AD versus control. The right graph shows the training and validation process. CNN demonstrated a very high accuracy at discriminating between AD and control on the basis of associated features and microglia features with very high areas under the curve. In summary, we have developed a cyclic fluorescent multiplex immunohistochemistry protocol that helps unveil the complexity and diversity of associated and microglial reactions. We identify at least 3 phenotypes or states, and we believe that the intermediate phenotype could be either transitional or compensatory. The deep learning convolutional neural networks on associated and microglial features allowed us to classify AD and control brains with high accuracy. Thank you very much for your attention.
Hi, my name is Mei Cheng. I'm a postdoc fellow from the IU AD Center, and I'm going to talk about the functional brain networks in AD and its relation to gene expression profiles. So we have known that AD mainly targets the hub regions in brain networks. For instance, the hippocampus in the medial temporal lobe and precuneus in the default network, and some hub regions in the visual cortex. But we don't know why those hub regions are selectively vulnerable in AD. Recent work have shown that the gene expression patterns of a few genes are related to the A-beta and tau accumulation patterns. For instance, the mapped gene showed strong gene expression in the medial parietal and also the medial temporal lobe, and the tau accumulation also showed a strong accumulation pattern in these brain regions. So we think that the gene expression levels may also shape the structure and functional connectomes in AD. So in this analysis, we aim to uncover the molecular mechanism underlying specific functional brain network changes in AD, and we hypothesize that the regional gene patterns would show specific spatial correlations with functional brain networks along the AD continuum. So we scanned the functional MRI in 47 healthy control, 46 SCD, and 34 MCI and 22 AD participants. We also used the gene data from the Island Human Brain Atlas. So in our sample, AD subjects have younger age than the other 3 groups, and we also have more females than males. So we computed the spatial correlations between gene expression patterns and brain connectivities. We selected 51 AD risk genes, and we found that the ECHDC3 gene showed the highest correlations to the brain connectivity, and the correlations increased from CN to AD. And also the other genes, so the HS3, ST1, also showed the strongest negative correlations in the 4 groups, and it's also increased from CN to AD. If we look at the spatial patterns for the connectivity and the gene, we can really say the 2 genes are correlated with the brain connectivity patterns. And these results are consistent with the results from one meta-analysis, which also identified the same 2 genes, and also the same 2 genes showed the inverse transcript patterns in AD. We then did individualized gene to connectivity analysis, and we replicated the results from the group level analysis. And we also did the analysis in the early onset AD and late onset AD, and we found that gene to connectivity correlation results are mainly driven by the early onset AD. Because some other genes also contribute to the brain connectivity, so we next construct a gene co-expression network and identified 5 modules, and we computed the spatial correlations between the genes expression levels from the 5 modules to the brain connectivity. And we still found 2 modules which show the inverse correlation patterns across the 4 groups. We also did a gene set enrichment analysis and found a few gene groups are enriched in the analysis. So in summary, we identified 15 AD genes which showed significant gene to connectivity associations, and we also found 2 of the 5 gene modules showed correlations. And we also identified the pathways which are positively enriched or negatively enriched. We are working on replicating the findings using the ADME data, and I need to thank my colleagues from IU and my collaborators from USC and UPenn and also NIH funding. And I also need to thank support from my family, and so my wife and my 1-year-old daughter. Thank you for your attention.
Today, I'm going to talk about my preliminary work on the increased cost of cognitive effort in Alzheimer's disease. Observational and clinical trials of Alzheimer's disease require accurate measurement of true cognitive performance. To do this, we administer neuropsychological test batteries that are cognitively demanding. The tests are difficult to provide some sensitivity to preclinical levels of disease. Given equal levels of reinforcement, individuals prefer less cognitively demanding activities. The magnitude of that preference correlates with performance on other cognitive tests. So if you have a very strong preference for a less demanding activity, you're going to perform less well on external cognitive measures. Older adults tend to find the same cognitive tasks subjectively more effortful compared to younger adults, even after you equate for overall performance. Finally, the computation of cognitive effort may be related to the accumulation of amyloid. So in this study, we wanted to test the hypothesis that individuals with preclinical Alzheimer's disease would find the same cognitive tests to be subjectively more effortful than cognitively healthy control participants. We also hypothesized that an objective measure of cognitive effort would outperform subjectively reported questionnaire-based measures, such as the need for cognition scale or the NASA task load index. As our objective measure, we use the cognitive effort discounting paradigm developed by Westbrook and colleagues. It consists of 2 phases. Training on a cognitively demanding task, we use the NACC in our version, and then an effort discounting paradigm. And I'll show examples of both of those. In the NACC, you're shown letters on the screen one at a time, and you have to indicate if the letter that's currently on the screen is the same as the letter that was shown N trials back. So in the 1 back, you're monitoring if the current letter is the same as the previous letter. In the 2, 3, and 4 back, you're monitoring whether the current letter is the same as 2 ago, 3 ago, or 4 ago, respectively. This task gets phenomenally difficult as you increase the level of letters that they have to keep in mind. Participants in our test did 2 minutes of each of these levels. Then in the discounting phase, we present choices. We present monetary rewards for completing certain levels of the test again. So one example here would be, do you want to receive $1 for completing the 1 back or $2 for the 2 back? And they simply choose which they preferred. If they chose the lower reward, we titrate the offered reward down by a certain amount. And if they choose the higher reward, we titrate it up by a certain amount. And we keep titrating the levels on offer until the indifference point is reached. The amount of money that would be equally preferred for the 1 versus 2 back, or the 1 versus 3, or the 1 versus 4, and so on. We normalize this indifference point to a subjective value, which is simply the highest money selected divided by the highest amount. So the thing to keep in mind is that a higher subjective value rating corresponds with lower cognitive cost. We wanted to see if this subjective value measure was sensitive to the accumulation of amyloid. So we collected data on 67 participants from the Knight Alzheimer's Disease Research Center who had CSF measures of amyloid. And the data are presented here. The subjective value is on the y-axis, and the levels of amyloid, the A-beta 42 to A-beta 40 ratio, is plotted on the x. And there is a significant relationship between these 2 measures, such that your subjective value or your cognitive cost increases as your levels of amyloid, your amyloid ratio decreases. So as you have a higher level of pathology, your subjective value is decreasing, or conversely, your cognitive cost is increasing. And this relationship is significant. When you look at the same CSF measures against the questionnaire-based reports, you do not see the same relationship, suggesting that our objective measure is indeed more sensitive than the questionnaire-based metrics. So some easy conclusions. This SV metric is sensitive to levels of amyloid in the cerebrospinal fluid. This is separate from overt performance on the NBAC task. Once you factor out NBAC performance, this relationship still holds. And finally, questionnaire-based metrics of cognitive effort do not correlate with amyloid. I want to take a moment to acknowledge my funding sources and mentors and collaborators, and of course the NIGHT ADRC participants and their families. Thank you for your time.
Hi, my name is Bistro Yortanova from University of Pittsburgh, and I want to tell you a little bit about a project in my lab about vascular contributions to Alzheimer's disease. About 2/3 of the people in the United States that have Alzheimer's disease are women. And in addition to the neuronal pathology and the neuroinflammation, in the latest years, people also agree that there is a vascular pathology of hemorrhaging and a local microinfarct and changes in the blood flow. So the vascular dysfunctions are present in both men and women, but the presentation appears to be sex-specific. So in my lab, we tried to figure out what the biological substrate of these vascular contributions is to the development of the pathology. We work in mouse models of Alzheimer's disease, APP-PS1, and looking at them longitudinally through time as they age, we found that vascular amyloid accumulates quite robustly in the form of cerebral amyloid angiopathy, just like in humans. And we also look at those mice and how this pathology interacts with the neurons in the brain. So here you can see a 2-photon image where neurons are green, vessels are red, and the plaques and the CA is shown in blue. And we found that the neurovascular unit function correlates with the amount of CA and plaques in the area. We also found using metabolic imaging that the tissue is hypoxic, and the bright regions here show us that the oxygen gradient as it goes from the artery to the vein drops much faster in the Alzheimer's mice. What we also found is that the accumulation rate of the tissue plaques was somewhat linear as the mice started aging, but the CA, if you can see the y-axis here, is broken because the accumulation of CA becomes much more rapid in the older mice. And when we looked to compare males and females, we found that females had consistently higher rates of CA deposits in their arteries. And this was most pronounced in the cortex. So when we measured blood flow using MRI in the cortex of both model mice of Alzheimer's and controls, we found that females consistently, the pink region on this side of the Violin plots, had a lower blood flow. The difference, though, was not as large as we expected based on the CA accumulation. But then when we looked at the distribution of the blood flow across the cortical and subcortical regions, we can see that in control animals, the drop of the blood flow was somewhat homogeneous across the cortex, versus in the Alzheimer's mice, there was a sex-specific difference where the females were dropping much more in the cortex. So we quantified this across age, and we were surprised to also see that while the cortical blood flow was really dropping, in the thalamus, we were observing rise in the blood flow. And this was combined with increasing rates in the glucose metabolism in the females. So in summary, I told you that vascular dysfunctions are present in both men and women, but the presentations appear to be sex-specific. I also told you that AD mice display tissue hypoxia and decreased blood flow, and this is in original distribution in the brain, with cortex having much higher drops in the blood flow. AD mice develop CA faster, and the tissue plaques in older females have a higher load together with the CA. So I hope I convince you that establishing sex-specific risk factors for vascular dysfunction of AD can benefit all humans. And I want to acknowledge all the amazing people that had worked on this project and all the funding that we received over the years. Thank you.
Hi, my name is Elisa McMahon, and I'm a postdoctoral fellow at the Rush Alzheimer's Disease Center at Rush University Medical Center in Chicago, Illinois. Today, I'll be presenting on decision-making and diabetes in older African Americans. Diabetes is a chronic condition that affects millions of Americans and requires a lot of management, both independently and with a healthcare provider. African Americans are 60% more likely to be diagnosed with diabetes and twice as likely to die from diabetes than non-Hispanic whites. African Americans also have a higher prevalence of some of the risk factors for diabetes, including comorbid conditions such as hypertension and obesity, as well as having higher exposure to social determinants of health associated with diabetes and its risk factors, including socioeconomic status, such as level of education and income, and neighborhood segregation. Simply because diabetes is a condition that requires continual management, it is very important to understand diabetes risk and management for older adults. However, it is additionally important because it is a risk factor for more rapid cognitive decline and dementia onset among older adults. Further, African Americans with diabetes are at higher risk for dementia than non-Hispanic white older adults with diabetes. So the possible risk factor that we're looking at here is decision-making. Decision-making is a complex process which involves evaluation, comparison, and selection of an optimal choice among competing alternatives. We all make decisions all day, every day, from deciding what to eat, what to wear, how we'll commute to work. These are all decisions that affect and shape eventual long-term outcomes. Decision-making is a cognitive process as it involves these components of evaluation, comparison, and selection, but high cognitive function does not necessarily mean optimal decision-making. In older age, the importance of decision-making takes on higher significance, especially in the realms of health and financial decision-making. Some of the decisions that older adults are faced with that have significant consequences are selecting and navigating insurance and healthcare providers, retirement planning and management, and end-of-life health and financial planning. As people age, they become more vulnerable to poor decision-making. Related to health and financial decision-making, such vulnerabilities can include not taking advantage of healthcare and related benefits, inadequately planning for retirement, making poor investment decisions, and engaging in risky credit or spending behavior. There are still very few investigations of racial disparities in decision-making, but there is some evidence of variability by race. In healthcare, African Americans are less likely to have an advance directive, to appoint a durable power of attorney, or engage in healthcare-sponsored preventative services. They are less likely to participate in financial markets or investment opportunities, and more likely than whites to be victims of fraud and abuse. Our team at the Rush Alzheimer's Disease Center has documented many health consequences of lower decision-making ability, including increased risk for Alzheimer's disease, mild cognitive impairment, and mortality. And we are beginning to extend this work to look at the relationship between decision-making and health in African Americans. In this study, we ask, is there an association between decision-making ability and objective measures of diabetes indicators in African Americans? Our data is 328 older African Americans participating in a longitudinal aging studies with no dementia at baseline. The decision-making measure is 12 items resembling real-world health and financial materials. The diabetes indicators are blood-measured hemoglobin A1C and blood glucose. Covariates are demographics, including age, sex, and education, and risk factors for diabetes, including depressive status, symptomatology, hypertensive status, and known diabetes status. For analysis, we modeled sequential OLS regression separately for hemoglobin A1C and blood glucose as outcomes. First, we included demographics and then added risk factors for diabetes. And finally, we looked at an interaction model to see if any observed association varied by diabetes status. For all analysis, we did sensitivity analyses for cognitive function to ensure that any observation was not driven by low cognitive function individuals. We found that decision-making was not associated with blood glucose level. However, we found that high decision-making was associated with lower levels of HbA1c. In the interaction analysis, this association was only present in individuals with known diabetes. So in conclusion, among individuals with known diabetes, higher decision-making ability was associated with lower levels of HbA1c. High levels of HbA1c are indicative of glycemic instability over long periods of time. Controlled levels are a really good indicator of successful diabetes management. Diabetes management involves complex decisions, managing medications, healthcare, diet, and lifestyle. Among individuals with diabetes, interventions to increase decision-making abilities could help to maintain healthy levels of HbA1c. Thank you for your time. If you have questions, here's my email and my Twitter handle.
Hello, my name is Radhika Jaganathan. I'm a clinical fellow in behavioral neurology and a REC scholar at Columbia University. Today, I'll be speaking about my work studying transcriptional dysregulation in Parkinson's disease by ATF4 transcription factor heterodimers. In the study of neurodegenerative diseases, gene expression analysis has been utilized as a valuable tool to identify dysregulated cellular pathways that may contribute to disease onset and progression. However, the upstream mechanisms leading to pathogenic alterations in gene expression profiles remain incompletely understood. Transcription factors are key cellular components that regulate gene expression by interacting with DNA and other elements of the transcriptional machinery. ATF4 is a stress-responsive transcription factor that has previously been implicated in Parkinson's disease. My work has utilized a cell biologic and bioinformatics approach to dissect the effect of a specific ATF4 transcription factor heterodimer consisting of ATF4 and another transcription factor, CRB3L2, on PD-associated gene dysregulation. To confirm that these transcription factors are expressed and form a complex in Parkinson's disease, I performed a meta-analysis of 7 published microarray datasets for ATF4 and CRB3L2 expression, and found that both ATF4 and CRB3L2 are upregulated in the substantia nigra of PD patients as compared to controls. I also obtained paraffin-embedded midbrain sections from the Columbia University Brain Bank. I performed a proximity ligation assay, which is an antibody-based method to identify specific protein-protein interactions, to look for complex formation, and found increased formation of the ATF4 CRB3L2 heterodimer, as indicated by these red-brown puncta, in the nucleus of Th-positive neurons in the substantia nigra of PD patients as compared to controls. To determine the effect of this transcription factor complex on PD-relevant gene expression, I utilized an approach where I compared a list of differentially expressed genes in Parkinson's disease with a list of all possible transcriptional targets of the ATF4 CRB3L2 complex. The list of differentially expressed genes was derived from a published meta-analysis of microarrays performed on substantia nigra from PD and control patients. To identify the list of ATF4 CRB3L2 targets, our lab developed a new technique called ChIP-MERA, in which ATF4 and CRB3L2 are overexpressed in HEC293 T cells. Each overexpressed construct contains a special domain that can be induced to heterodimerize by addition of a chemical dimerizer. The resulting cells overexpressing the forced complex are subjected to chromatin immunoprecipitation sequencing to identify possible gene targets of ATF4 CRB3L2 regulation. I compared the list of differentially expressed genes with the list of ATF4 CRB3L2 target genes from the ChIP-MERA analysis to identify a network of 255 genes that are both dysregulated in PD and potential targets of the ATF4 CRB3L2 complex. I performed a string network analysis, which is a tool that allows one to determine whether a dataset is likely to be found randomly or if there are functional relationships between the nodes in question, to demonstrate that the majority of genes in this overlapping dataset are functionally related. I then performed a functional enrichment analysis on this network of overlapping genes. This analysis identified familiar terms known to be associated with PD pathogenesis, such as mitochondrial organization and regulation of microautophagy. However, I also saw enrichment of previously unidentified processes, such as mRNA processing and RNA splicing. Interestingly, while alternative splicing has been linked to other neurodegenerative diseases like ALS and Alzheimer's disease, dysregulation of RNA processing as a whole has not been systematically studied in PD, suggesting that this may represent a new avenue of exploration for understanding Parkinson's disease pathogenesis. This work demonstrates that the ATF4 CRB3L2 complex is upregulated in PD and may be involved in the regulation of disease-specific pathways. Importantly, it also suggests that this approach of studying a gene expression network through the lens of specific transcription factor activity may allow us to identify previously unknown disease-relevant pathways. Future work will focus on validating the results of this analysis using cells from conditional ATF4 and CRB3L2 knockout mice that we have generated in confirmatory in vitro assays. With that, I will wrap up. I'd like to thank my lab mentor, Dr. Hengst, and my clinical mentor, Dr. Martyr, Thanks also to all of my lab mates and Dr. Small and Dr. Cosentino of the Columbia ADRC. My contact information is on this slide if anyone would like to reach out with questions. Thanks so much.
Hi, I'm Adam Mecca from the Yale ADRC, and I'm going to review some of our recent work that closely examines the relationships between synaptic density measured with PET and cognitive performance in Alzheimer's disease. For 30 years, synaptic loss has been referred to as the major pathological correlate of cognitive impairment in AD. However, the statement is based on a limited number of patients studied by autopsy or biopsy and a few brain regions at the moderate to severe stages of disease. The seminal papers in this field included brain biopsies of frontal cortex or associated postmortem synapse density with antemortem cognitive performance. A subsequent analysis by Sheff in 2006 studied the dentate gyrus in a pooled sample that included earlier stages of disease. And we've begun to evaluate synaptic alterations in vivo with SV2A PET imaging. Synaptic vesicle glycoprotein 2 is an essential component of synaptic vesicles and is located in presynaptic terminals. And one isoform SV2A is expressed in virtually all synapses, which makes it an ideal target that led to the development of the tracer UCBJ for quantitative PET imaging of synaptic density. In our previous work with UCBJ PET, we demonstrated widespread reductions in synaptic density in AD in the medial temporal lobe and neocortical regions. In this figure from our recent report showing average coronal images of synaptic density expressed as DVR, there's visibly lower UCBJ binding in the medial temporal lobe, but also throughout the neocortex and subcortex. In the present study, we focused on examining the relationship between in vivo synaptic density measurements and cognitive performance. The sample consists of 19 cognitively normal participants that were PIB negative. This group was used to compare participant characteristics and relative cognitive performance between groups. Correlations between synaptic density and cognition were examined in 45 participants with Alzheimer's disease, all of whom were PIB positive. And the AD group was divided between amnestic MCI due to AD and mild AD dementia. Their average age was about 71 years and global CDRs were 0.5 or 1. The neuropsychological test battery was administered to assess performance in 5 cognitive domains, and the AD group had significantly lower performance for verbal memory, language, executive function, processing speed, and visuospatial ability, as well as a combined measure of global cognition. I won't review the PET methods in detail, but briefly we calculated a distribution volume ratio using the SRTM2 and cerebellum as the reference region. In the 45 individuals with either MCI or mild dementia due to AD, global cognition was highly correlated with synaptic density in the composite of AD-affected regions. This multiple linear regression model controlled for age, sex, and education. Similar models were also fit with each cognitive domain as the outcome, and the effect sizes were similar with eta values between 0.45 and 0.5 for individual domains. And interestingly, the weakest effect is for verbal memory, which is likely due to a floor effect for the tests used to assess this domain in our sample. We wanted to investigate if these results were simply due to volume loss, so we performed the same analyses but after partial volume correction. The associations remain significant for global cognitive performance in each of the 5 cognitive domains. Importantly, this finding was robust to analysis method. We also examined the regional pattern of synaptic density that's correlated with cognitive performance. And for these analyses, the correlation coefficient for DVR and global cognition in each brain region is projected onto the FreeSurfer Atlas and overlaid on the template MRI. Significant correlations are shown with a color scale for strength of correlation. We can see positive associations broadly for cortical and a few subcortical regions. These relationships can also be corrected for atrophy as shown in the middle panel. Partial volume correction demonstrated very similar results to those observed in non-corrected data, with positive associations in almost all of the same cortical and subcortical regions, but with slightly reduced strength of association and regional extent. So again, convincing that there is an effect beyond that due to atrophy. And finally, we examined the association between brain volumes and cognitive performance. The pattern remains somewhat similar, but with still further reduced strength of association and regional extent. The correlation between synaptic density and cognitive performance is more robust than with brain volume. In summary, synaptic density was a significant predictor of global and domain-specific cognitive performance. The results confirm neuropath studies demonstrating a significant association between synaptic density and cognition in AD, and they also support the use of synaptic imaging as a potential biomarker outcome for therapeutic trials. Thank you very much for listening. I want to thank my mentors, co-workers, and collaborators, and especially the people who participated in this study.
Hi, I'm Dr. Anna Daugherty, and thank you for the invitation today to share some of my work about iron-related oxidative damage. In typical aging, non-heme iron accumulates outside of binding complexes to cause oxidative damage. This in turn promotes expression of pro-inflammatory cytokines. It increases the rate of apoptosis and it causes DNA mutations. This collection of mechanisms are proposed to drive the neural and cognitive declines that are typical in aging and in greater severity constitute Alzheimer's disease and related dementia. Throughout my work, I have applied MRI methods to quantify brain iron concentration as a proxy biomarker of oxidative damage. Here's an example of quantitative susceptibility mapping, or QSM. On this type of image, darker intensity value corresponds to proportionally greater iron concentration. Applying techniques like this in community samples spanning nearly the entire adult lifespan, I've shown that advanced age correlates with greater iron concentration in age-sensitive regions. In longitudinal study, greater brain iron accumulation predicts faster atrophy and precedes and predicts declines by nearly 7 years, as well as cognitive deficits. Here, for example, individuals with greater baseline iron concentration in the caudate experience steeper declines in verbal working memory over 2 years. I've shown similar relations between high levels of iron accumulation in the hippocampus, predicting declines in declarative memory. More recently, I've examined the relation of striatal iron concentration as a biomarker of oxidative damage in relation to other neuroimaging markers of Alzheimer's disease-related pathology. This is a cross-sectional study of middle-aged and older adults who are community dwelling and experiencing preclinical cognitive decline. And we identified a complex interaction between striatal iron concentration, whole brain SUVR, and age when predicting intrarenal cortex volume. Now, the intrarenal cortex, of course, is a region that is vulnerable to decline early in Alzheimer's disease. And so on the vertical y-axis, I'm illustrating the relation between intrarenal cortex volume and age, and negative values would indicate for an accelerated rate of atrophy and possible increased risk for Alzheimer's disease. On the horizontal bottom axis is R2 star, and higher levels would indicate greater iron concentration. And here, for example, is a negative correlation. We examined that across levels of SUVR, moving from left to right, low, moderate, and high based off of clinical criteria. And at increasing levels of SUVR, we can see an exacerbated effect of iron concentration, so that individuals who had both high iron concentration and high SUVR demonstrated smaller intrarenal cortex volumes with age. And this is a cross-sectional study and we can't infer causality, but it does suggest a confluence of risk factors that together may signal a profile of individuals who will develop Alzheimer's disease later. When examining this risk profile, if you will, I started to think about the common causes and antecedents that may influence early and midlife health that would set an individual on a trajectory towards risk for Alzheimer's disease. And I naturally led to cardiovascular health. Now, iron homeostasis is dependent upon endothelial function, and we know that midlife hypertension increases risk for Alzheimer's disease almost twofold. Importantly, blood pressure control does not mitigate this risk, and so my current work is examining alternative pathways that may convey vascular risk effects on Alzheimer's disease pathology, with particular emphasis on iron-related oxidative damage, cerebral microbleeds, and pro-inflammatory cytokines. Most importantly, this risk disproportionately affects Black and Hispanic elders who are more likely to be diagnosed with hypertension and less likely to achieve blood pressure control as compared to white counterparts. And so examining these pathways and identifying means to mitigate risk are essential for promoting the health and well-being for our diverse populations. This is the intent behind my current work at the center in Michigan, where we are examining community-based samples with consensus diagnosis and following them longitudinally with several different blood imaging, biomarkers, and cognitive assessments. And this work is currently funded by a pilot award in the center. With preliminary data, I can share that elevations in pulse pressure as an index of endothelial function indeed correlate with higher iron concentrations in the caudate and hippocampus, and this work is ongoing. Thank you for your time and attention. Please reach out with any questions, and have a good day.
Well, I just want to say that was a wonderful session. Those talks were so enjoyable. It's really wonderful to see all of the great science that's happening across the nation. This wraps up our research education component session.